just now we have talked about the pentose phosphate pathway, right? And this pentose phosphate pathway is another important pathway. And here you should remember that in these pathways, they have generated two very important molecules, NADPH and ribose 5-phosphate. But in these pathways, there's no ATP can be generated. And this pathways is also like glycolysis also takes place in cytoplasm, okay? And the reaction of these pathways can be divided into two phases. The first phase is irreversible oxidative phase, and the second phase is irreversible non-oxidative phase. And you should take you should pay attention to the first phase, the irreversible oxidative phase. Okay. And in the first phase, as you see, they started with the glucose 6-phosphate, right? And then they can be converted into the ribose 5-phosphate. And during these reactions, the first step is a rate-limiting step, and this step is catalyzed by the glucose 6-phosphate dehydrogenase. Also we call it as a G6PD. And this enzyme, you should remember, this is a key enzymes involved in the pentose phosphate pathways. And here you should notice that during the first phase, the NADPH can be generated, right? And then the ribose 5-phosphate, these two important molecules can be generated from the oxidative phase. Okay, and then next, the second phase is a reversible non-oxidative phase. Here, in this phase, it can be involved a series of group transform reactions, and after a series reactions, the ribose 5-phosphate can be converted to the fructose 6-phosphate and glyceraldehyde 3-phosphate. And these two molecules in fact are intermediates in the glycolysis. So these two molecules can be re-entered to the glycolysis pathways. Okay, so this is the reaction of pentose phosphate pathways. And how to regulate the pentose phosphate pathways? In fact, it depends on the activity of these enzymes, glucose 6-phosphate dehydrogenase. And the activity of these enzymes depends on the need of the NADPH in our body. If we need more NADPH, and then this enzyme will be activated. If we don't need NADPH, and then this enzyme the activity of this enzyme will be inhibited. Okay, so this is the regulation of the pentose phosphate pathways you should remember. In fact, the regulation of these pathways is to regulate the activity of this G6PD. And the activity of these enzymes depends on the need of NADPH in our body. Okay. And next, you should remember the physiological significance of pentose phosphate pathways. And the pentose phosphate pathways provide two very important molecules you should remember. The first one is ribose 5-phosphate, because the ribose 5-phosphate is very important. Is very important source for the synthesis of nucleotide or nucleic acid, right? And another very important molecule is NADPH. NADPH is very important. NADPH can be acted as a reducing power for the synthesis of the fatty acid or the cholesterol and so on. And also NADPH can also act as a antioxidant molecules. So this is you should remember. And next, we have talked about the G6PD deficiency. This is a genetic disorder. Okay. Next, I will show you a video. These videos introduce the G6PD deficiency counts, and maybe from some certain drugs maybe cause the severe hemolytic anemia. Okay, so let's take 10 minutes break. Okay. Can you see the screen? Can you see the screen? Is everything goes well? Okay, now next we will talk about the glycogen synthesis and catabolisms. Glycogen is a major storage polysaccharides in animals. It's similar like the starch in plants, and it's a branched polymer of alpha-D glucose, but a more highly branched structure than the amylopectins. And here, okay, stop here. Is that okay? And here from this picture shows you the structure of the glycogen. As shown in this figure, instead of being one long chain, it consists of many short chains cross-linked together, right? And the glucosyl units of the chains are alpha-1 and 4 glycosidic bonds. But the cross-linking between chains is by alpha-1 and 6 glycosidic bonds. And the glycogen chains has only one reducing end here, has one reducing end, but has lots of non-reducing end. Here is non-reducing end. This is also non-reducing end, and depends on the how many branches it has. Okay, and after meal, when the blood glucose concentration and the insulin are higher, the glucose is mainly taken up by liver and muscle and used to replenish their glycogen stores. Glycogen occurs mainly in liver and muscle, which is not just stored appreciably in any other cells in the body. First, let's see the synthesis of glycogen. The glycogen synthesis is also named as glycogenesis. The synthesis of glycogen is from the glucose, and the glycogenesis is an anabolic process. That means it requires energy. Okay, so next, let's see the reaction involved in the glycogenesis. When the G means the glucose, okay? When the glucose enters the cell, it's phosphorylated by ATP. And this reaction is catalyzed in brain and muscle by the enzymes hexokinase. HK means the hexokinase. And this kinase can transform the phosphorus group from ATP to glucose. But in liver, there is a different enzyme catalyzing the same reaction. In liver, the reaction called the glucose kinase. The GK means the glucose kinase. And the glucose kinase is essentially active only at a high glucose concentration. That means in fact, the hexokinase and glucokinase, they are isoenzymes, right? They can catalyze the same enzyme, same reaction, but they are translated from the different genes. And hexokinase, glucokinase, just now we said hexokinase is mainly located in the brain and muscle, but glucokinase is located in the liver, and they have the different affinity. They have different affinity to the glucose. The hexokinase has a high affinity to the glucose, but the glucokinase have very low affinity to the glucose. That means the Km value of the glucokinase is higher than the hexokinase, right? So this is very important to maintain the blood glucose level. Okay, and then the phosphoryl group can be switched from the 6 position to the 1 position by a second enzyme. This enzyme we named as mutase, phosphoglucomutase. And then next, the gluco-1-phosphate is converted into the activated form, UDPG. You should remember. UDPG means the uridine diphosphoglucose. And UDPG, it can be act as an active pattern of glucose, or can be act as an active donor of glucose in the glycogenesis. And in glycogenesis, UDPG is made by a reaction between UTP and glucose 1-phosphate. And the enzyme for this reaction is called UDPG pyrophosphorylase. And UDPG is activated or reactive glucose compounds that can donate its glucosyl group to the glycogen. Okay, so we move to the next slide. Glycogen synthesis occurs by enlarging pre-existing glycogen molecules. That means glycogen synthesis cannot happen into free glucose in our body. It should be occurs by enlarging a pre-existing glycogen molecules, and we call these glycogen molecules as primers or the glycogen primers. And this picture shows you how to form these glycogen primers. The initial steps in the glycogen synthesis involved a protein glycogenase. The glycogenase, in fact, is a protein that is glucosylated on a specific tyrosine residues by the UDPG. The glycogen catalyzes the transform of a further 7 glucose residues from the UDPG, and then forming a 1 and 4 linkage to form a glycogen primers. And this glycogen primers is a substrate for the glycogen synthesis. Okay, as you see here, these glycogens can transform glucose residues from the UDPG. So how many glucose residues from the UDPG is 7? This glycogen can transform 7 glucose residues from the UDPG. And this glucose residues is linked by the 1 and 4 glycolytic bonds. and then form this glycogen primer. Okay, and this glycogen primer can be act as a substrate for the glycogen synthesis. And next, after forming the primer, the synthesis of the glycogen is used UDPG as a glucosyl donor, and this reaction can be catalyzed by the enzyme glycogen synthesis. And glycogen synthesis catalyzes the formation of the glycoside bond between carbon 1 of the glucose and of the UDPG and the carbon 4 of a terminal glucose residues of glycogen. And then in this reaction, after the reactions, the UDP can be liberated or released. And then additional of the glucose residues to a glycogen chain or primers occurs at the non-reducing end, out end of the molecules, so that the branches of the glycogen molecules become elongated as the successive 1 and 4 linkage are formed. So the problem is that if more and more glucosyl groups was added to these primer molecules, the result would be very long polysaccharide chains. But as you know that the glycogen has a branch structure, right? So instead of consisting of long chains of glucosyl units, glycogen is a highly branched molecule. So how to form the branched structure? Okay, so this structure is achieved by another enzyme we call branching enzymes. Once the glycogen synthesis has made a straight chain extension more than 11 units in length, and then the branching enzymes transform a short block of terminal glucosyl unit from the end of an alpha-linked chains to the hydroxy group of carbon 6 of a glucose unit on the same or another chains. Okay, here. Then it creates more and more non-reducing end, both for the glycogen synthesis and breakdown. Okay, so this is a process for the glycogen synthesis. Next, let's see the glycogen catabolisms or glycogen breakdown. We also named as glycogenolysis. And the liver, as mentioned, as a store can store glycogen, but which is not used. But the glycogen stored in the liver is not used by the liver itself, but by the other tissues, and especially the brain and erythrocyte, because these two tissues, brain and red blood cells, cannot use the lipid as a source of energy. In fasting, such as between meals, it degrades glycogen and releases glucose into the bloodstream. So this allows the brain to take up glucose and continue to function normally. So next, let's see the reaction involved in the glycogenolysis. Glycogenolysis is not the reverse of glycogenesis, but is a separate pathway. Okay. As with synthesis, glycogen breakdown occurs at non-reducing end, and the glycogen phosphorylase catalyzes the rate-limiting step in the glycogenolysis. The phosphorylated cleavage of the 1 and 4 linkage of glycogen to yield the free glucose 1 phosphate. And there are different isoenzymes of glycogen phosphorylase in liver, muscle, and brain. And these isoenzymes encoded by the different genes. And this glycogen phosphorylase, is a key enzyme in the glycogenolysis. And next, the debranching enzymes involved in the removing branches from the glycogen. And the terminal glucosyl residues from the outermost chains of the glycogen molecules are removed sequentially until approximately about 4 glucose residues remains on either side of the 1 and 6 branch. And this figure shows you how the debranch enzymes involved in the removing branch from the glycogen. And here, as you see here, this branch points of the glycogen create a problem. That is, the problem is that the glycogen phosphorylase, cannot function with 4 glucose unit of such point because the enzyme is big, and the branch gets in the way of its attaching to the site of the glucosyl bond to be attacked. So how to solve this problem? and the debranching enzyme can take care of this problem so that the degradation can continue. So next, let's see how to solve this problem. The debranching enzyme has two catalytic sites in a single polypeptide chain. One is a glucagon transferase. The transferase that can transform a trisaccharide unit. This indicated by this yellow, okay, can transform the trisaccharide unit from one branch to the 4-hydroxyl group of another chain, and then exposing the 1 and 6 branch point. And this makes them part of a chain. This makes them part of a chain long enough for phosphorylase to work on. And the other active enzyme is 1,6-glycosidase activity. And 1,6-glycosidase that can catalyze the hydrolysis of the 1 and 6-glycoside bond to liberate free glucose or to release the free glucose. So, and then the first phosphorylase can be hydrolyze the glucosyl group. Okay. The combined action of the phosphorylase and these debranching enzymes leads to the complete breakdown of the glycogen to the glucose 1 phosphate. Okay, and then the glucose 1 phosphate is then converted by the mucus to the glucose 6 phosphate. And here you should remember, in liver, not in the muscle. The glucose 6 phosphate can be hydrolyzed by the glucose 6 phosphatase to give the free glucose, and then they can be released into the blood, which leading an increase in the blood glucose concentration. And this enzyme is very important. Glucose 6 phosphatase is in the lumen of the smooth endoplasmic reticulums, and the genetic defects of this glucose 6 phosphate transporter can cause a variant of type 1 glycogen storage disease. Okay, and then just now we have said in liver, because they have the glucose 6 phosphatase, so they can give to the free, they can catalyze the glucose 6 phosphate to the glucose and give the free glucose. How about in the muscle? The muscle cells, in the muscle cells, they have very low activity of the glucose 6 phosphatase, so they cannot convert it the glucose 6 phosphate to the glucose. So the glucose 6 phosphate from the muscle glycogen can be metabolized by the muscle itself and by entering the glycolysis pathways. Okay, so this is a catabolism and synthesis, glycogen catabolism and synthesis. So next, let's see the regulation of the glycogenolysis and glycogenesis. So there are two key enzymes involved in the controlling the glycogen metabolisms. The first one is glycogen synthesis, and the second one is glycogen phosphorylase. And the glycogen synthesis is involved in the glycogenesis, right? And the glycogen phosphorylase involved in the glycogenolysis. And these two enzymes can be regulated in opposite direction by allosteric mechanisms and covalent modification by reversible phosphorylation and dephosphorylation. For example, here the phosphorylation of glycogen phosphorylase can increase its activity, but phosphorylation of glycogen synthesis can reduce its activity, right? Okay. So last, let's summarize the glycogenolysis and the glycogenesis. And in fact, glycogen is the principal storage carbohydrate in our body, and mainly in the liver and muscle. But the glycogen in liver and muscle, they have different functions, right? Because, for example, in the glycogen in the liver, the main function of the glycogen in liver is to maintaining the blood glucose level. That is because why they have the different roles. That is because depends on the activity of the enzyme is the glucose 6 phosphatase. In liver, the roles of glycogen is to provide free glucose for export to maintain the blood concentration of glucose. But in muscle, the role of glycogen is to provide a source of glucose-6-phosphate for glycolysis in response to the need for ATP or in the for muscle contraction. Okay, and the glycogen is synthesized from the glucose by the pathway we named as glycogenesis. And there are 3 enzymes involved in the glycogenesis. Glycogenesis. There are 2 enzymes involved. I made a mistake. Okay, this is a glycogenesis. The left one shows you the glycogenesis, and the right picture shows you the glycogenolysis. And glycogenesis is glycogen is synthesized from the glucose by the pathway we named as glycogenesis. And there are 2 enzymes involved in these pathways: glycogen synthesis and branching enzymes. And it's broken down by a separate pathway we named as glycogenolysis. And there are 3 enzymes involved in the in the breakdown pathways: the phosphorylase, glycan transferase, and the debranching enzymes. Okay, and these 2 enzymes, glycogen synthesis and phosphorylase, are key enzymes involved in the glycogen metabolism. Okay, so